Hello, very good morning to all of you. Dear friends, welcome back to the video lecture series. We are discussing here the analysis of food, section first of this CHA 393B paper, and our current chapter is chapter number 6, and it is related with the protein. Okay, so protein component of the food material, and in this protein chapter there are two sections. Section A is related to the protein analysis, and second section, that is B section, is related to the protein characterization. We were discussing protein characterization. In last lecture we have seen the amino acid analysis, then protein nutritional value, okay, that is PDCAS method, and one more method. Okay, now there are other methods also which are essential or which are done for the protein nutritional value or protein nutritional quality checkup. That methods are utilized here. The concept is essential amino acid index. Essential amino acid index, the short form is EAAI, essential amino acid index. Actually, this essential amino acid index estimates the protein nutritional quality. Protein nutritional quality means the nutritive value of the protein based on content of all essential amino acids, based on content of all essential amino acids compared to reference protein, okay. So this method or this concept is based on mainly the content of all essential amino acids compared to reference protein. This EAAI method is a rapid method. It is giving very fast results for the evaluation of amino acid content of food formulations. Means different food products are there, and we can evaluate the amino acid composition, amino acid content of all essential amino acids, let us say. Okay, means the PDCAS method that we were discussed earlier, that was related to the estimation of, or it is concerning with it accounts for only first limiting amino acid. In PDCAS method we are analyzing the first limiting amino acid only. But in this EAAI method, this method is accounting for the all essential amino acids. So this is the major difference between the EAAI method and PDCAAS method. Okay, so what is the main difference? The PDCAS method is considering only first limiting amino acid, okay, in the food content. But the EAAI method is considering all essential amino acids present in the given protein or food. But there is one drawback of this method, and that is related to this EAAI method does not estimate protein digestibility at all. Okay, though it is considering all essential amino acids, but this method is not considering the protein digestibility factor, because this protein digestibility factor is also important from the nutritional quality point of view of the protein. Hence, This is one of the drawback of this EAI method. This EAI index, that is essential amino acid index, is calculated by using this formula. We know that there are only 9 essential amino acids required for the human body. Okay, the list that we were discussed in the last lecture, that is lysine, histidine, tryptophan, valine, etc. One more, total 9 amino acids are there. These are called as essential amino acids, and these are required in our diet. These amino acids must be there in our diet, in our food. Okay, so that our body could because our body cannot synthesize rapidly these amino acids in our body. Okay, so that's why they are needed in our diet. So this essential amino acid index is calculated by using a formula. So this is not a square root, this is a 9th root. Why it is 9th root? Why it is not square root, cube root or 4th root, etc.? Because we know that essential amino acids are 9 in number. Okay, 9 in number, and hence this is a 9th root. Okay, for calculation purpose, this is 9th root of all the data of 9 essential amino acids. Okay, and this is a multiplication of all data of all essential amino acids, and the data is, this is the ratio of milligram of lysine. Lysine is taken for example, only for example, because you can take the name of any essential amino acid here. Okay, and the remaining 8, same ratio we have to take for the remaining 8 amino acids. Okay, in this 9th root, this is the under root of this under root sign. Okay, so what is the ratio? Milligram of lysine in 1 gram of test protein. Milligram of lysine means milligram of that one essential amino acid in 1 gram of test protein, divided by milligram of that lysine here, or that specific essential amino acid in 1 gram of reference protein. Okay, so this ratio is for 1 essential amino acids. So we have to multiply all these ratios for remaining 8 amino acids. There are 9 essential amino acids, so this ratio is for only 1 essential amino acid. We have to multiply this by for remaining 8 essential amino acids. Okay, same ratio, milligram of tyrosine, milligram of tryptophan, milligram of valine, milligram of etc. etc. Okay, so this is the ratio, and the multiplication, the answer, multiplication of all these 9 essential amino acids, the ratios of all these 9 essential amino acids. Okay, then 9th root is taken, and that is known as essential amino acid index, EAI. Okay, now the second concept in this, in vitro protein digestibility. As we know that in this case the protein digestibility is not considered, hence in vitro protein digestibility is another method. It is called as pH shift method. pH shift method. Okay, so this is in vitro protein analysis. In vitro means here we are not using animals, or we are not using any organism in this case. So that's why here is we are doing a chemical reaction, and that's why this method is called as in vitro protein digestibility pH shift method. In vitro means there is no use of any organism, but by using a physical or a chemical method we are going to do analysis or assay. Okay, so this is in vitro protein digestibility method by measuring extent of protein hydrolysis. So how to do this method? This is done by measuring extent of protein hydrolysis. So protein hydrolysis, how much protein hydrolysis has been done, has been occur, that extent of protein hydrolysis upon a reaction, upon set up a reaction under standardized conditions, with commercial digestive enzymes, okay. So with the help of commercial digestive enzymes, a reaction is set up with the protein hydrolysis, okay, and the conditions are standardized. So this method is called as pH shift method, okay, and this is protein digestibility in vitro method. The pH shift method combines, actually there is a combination of two factors or two calculations. The one is EAA composition, that is essential amino acid composition, and calculation based on in vitro assay of digestibility. So these two combinations, these two calculations are combined together, and this gives a data by the pH shift method, okay. The protein efficiency ratio calculation, okay, and the protein efficiency ratio calculation is intended for routine quality control analysis, okay. So this is the part of this C pair, that is calculation of protein essential ratio is the part of this pH shift method, okay, and this is done for routine quality control analysis in the food processing industries, okay. Now the next point is lysine availability. So what is lysine availability? Here lysine is the name of one of the amino acid, and in some food processing units we are going to use heating processes, okay, and in such heating processes the lysine is not properly, or the nutritional value of a heat processed product is lower than the expected one, okay. So one should assess the availability of lysine in the product, okay. Means if the nutritional quality of a heat processed product, if it is lower than the expected, then the expected value of that lysine availability should be calculated, okay, and hence lysine availability must be taken into consideration. The free epsilon amino group present in the amino acid structure is responsible for the reaction with most of the components present in the food, okay. So this NH2 group present at the epsilon carbon atom in the given structure of that amino acid is responsible for the reaction with different food components present in that food formulation along with the during this heating processes or during food processment in the food process units, okay. So lysine can readily complex with reducing sugars in the browning reaction and oxidize polyphenols, okay. So hence the method to measure available lysine is a spectrophotometric method, So by using spectrophotometric method we can calculate this availability of lysine by using a reagent known as 1,fluoro-2,3-dinitrobenzene. 1,fluoro-2,3-dinitrobenzene. So this reagent is used for the spectrophotometric determination of lysine availability. Okay, so this can be used to determine if food processing operations have reduced the availability of lysine in the food. Okay, so when we are processing the food formulation for different purposes, then the availability of lysine is get reduced, and hence the lysine availability should be determined by using this method, and the method is spectrophotometric method, and the reagent used is 1,fluoro-2,3-dinitrobenzene. Okay, so this is all about the remaining three topics related to protein nutritional quality. Okay, first one is EAAI method, second one is in vitro protein digestibility, that is PS shift method, and the next one is lysine availability. Okay, thanks for watching this video. Have a nice day.